Right. Thank you for the opportunity to share our work on real-time metabolic profiling of pyruvate to lactate conversion in mammalian cells using SABRE. And of course, I want to thank my PI, Professor Chekmenev, for his invaluable and continuous training that have led to the success of this lab. Oops, what's happening here? One second, I don't think. Yep. I also want to acknowledge Kamanos Cancer Institute, a cancer research facility in the medical school at Wayne State University. This facility made access to cancer cells and space for us to move our instrumentation in to perform this experiment. And of course, I want to appreciate all the people who have made impact in this research, especially Dr. Tom Horn, Professor Yubin, Dr. Su, all my lab members and other collaborators, not forgetting our active funding sources, NSF, NIH, DoD, and DOE. Let me quickly show you around the overall workflow for the benchtop analysis of cellular metabolic flux, the details of which Kamal, who will be presenting right after this talk, will cover from sample preparation to how we obtain these results. So here we have cells being prepared and incubated in NMR tube at 37 degrees Celsius in parallel with the preparation of the SABRE molecule. So after several cycles of paradigm through the SABRE sample, we obtain maximum polarization, and then we process the sample and homogenize it in the cells in the NMR tube sitting on the benchtop spectrometer. And once we have pyruvate in cells, it converted into several other downstream metabolites that could be spectroscopically measured and quantified. So these are typical examples of spectra that could be obtained from such an experiment. For biocompatible preparation, we first polarize the sample and check for 8 polarization using C13 NMR spectroscopy. And once you are satisfied with the polarization, we pick the sample into a syringe containing butyl acetate dosed with few millimolar Sudan blue dye, which create two distinct phases, the organic and then the aqueous phase. So the aqueous phase containing the hyperpolarized material is taken into another syringe containing a buffer, which is transferred into a test tube sitting on an elevated water bath, and a gas is blown through to evolve residual solvent, leaving our hyperpolarized material, which is in a biocompatible solvent. And then we also check for 8 polarization, after which this material could be used for any biological application. And of course, I want to mention that the entire process for the biocompatible preparation is guided in 200 millitesla field because of the favorable T1 of pyruvate in this field compared to what we observed in the Earth magnetic field. So where do we stand with this biocompatible preparation? In the month of September, when we piloted this experiment, for a typical 70 millimolar nominal concentration of pyruvate load, we were seeing something around 30 millimolar residual pyruvate concentration and a final acetone concentration of around 10 millimolar, which we expose our biological specimen to. And this concentration is several orders lower than the FDA approved limit, with the substrate concentration, substrate polarization around 5%. So we validated this method in yeast cells, and these are typical spectra that could be obtained for such an experiment. And here, our main goal was to monitor for two key downstream metabolites, which are carbon dioxide and then bicarbonate. So once you have pyruvate in yeast cells, it is primarily converted into CO2, which is further converted into bicarbonate. So with the pyruvate in the yeast cell, we saw spectroscopically the signal of the pyruvate decaying gradually whilst we steadily build up our downstream metabolite, and eventually they also decay due to relaxation dynamics. So after we have validated and then confirmed that everything was working, we now transition to more sensitive cells. So here the experiments were done in leukemia cancer cells. And here the goal was not to monitor for bicarbonate and then carbon dioxide, but metabolically monitor the pyruvate conversion to lactate. And also we performed several other assays to be able to correct for polarization of pyruvate, the concentration, and then the number of cells performing these metabolic activities. After which we were able to extract a nice dynamic lactate curve as against the pyruvate decay signal. So after we're able to perform all these normalization, we can now start the metabolic profiling, which is very key for us to be able to delineate cells. So from the downstream metabolic dynamic curve here, we are able to reach peak lactate and KPL, which is in long-term effective pyruvate to lactate conversion rate. And these parameters could be plotted independently for the metabolism of the individual cells. And here we were dealing with HL60 cell lines, but we had HL60 parent cell line and another HL60 cell line, which is chemo resistant. And we could be able to, or we were able to delineate their metabolic profiles, and we could see very strong statistical significance from both the peak lactate and then the KPL, which well agrees with the Agilent Seahorse data that was provided by our collaborator with statistical significance between the resistant cell line and then the parent cell line, meaning whatever we are seeing with our technology matches with an already existing method. And instead of dealing with these parameters individually, we can as well plot multi-parameter or principal component analysis with one dimension being peak lactate and then the other, the KPL, and plot these data to create a 95% confidence limit to delineate these cells. What else can we do? Here we had three different cell lines, which were all parent cell line, the U937 parent cell line, HL60, and then MV411 cell lines. And we could differentiate their metabolic profiles in triplicate experiment, and we could see a clear and strong statistical significance from both their peak lactate and then the KPL as well as the principal component analysis. Well, here we didn't have a 95% confidence level because of the data size, but in the other cells we could see a 95% confidence level with clear delineation between their metabolic profiles. And again, we had breast cancer cells to delineate their metabolic profiles. So we were dealing with a metastatic 4001 breast cancer cell and then a non-metastatic 4007 breast cancer cell. And again, you could see clear statistical significance between these two cell lines from their peak lactate the KPL as well as the principal component analysis. Here we had slightly different metabolic preparation, meaning we tried to reduce the concentration of the pyruvate that we were injecting to the cells to something low in the order of 1 millimolar compared to the 30 millimolar that the cells were previously exposed to. And the experiment was done in primary brain cells. And yet we were able to see significant levels of lactate from the convention of pyruvate. And here the key interest was not in cancer, but newly generative diseases, which is in the interest of the lab that provided the cells. We also wanted to investigate how low we could go with the levels of pyruvate that we can inject into cells. And here we started with something low in the order of 180 microliters of initial pyruvate injections. And still, we were able to measure significant levels of lactate from our experiment, which is an outstanding on ketone with a very nice downstream metabolic curve. So in summary, our long-term goal is to be able to obtain specimen from blood or maybe biopsies or any other means to be able to perform low-field NMR analysis to better help in the diagnosis, treatment, monitoring, risk assessment, prognosis of not only breast cancer, but applied to many other kinds of cancer. And with this, I would like to thank you all for your attention.
Thank you so much. Beautiful work. As always, if you all have questions, feel free to put them in the chat. Yeah?
We have time. We don't have time for one.
We do have time for one. You want to go for one?
I do. Joseph, this is really beautiful work. A question about the chemotherapy resistance measurement. So if I understand the measure, well, can you explain the experiment to me? So it seems like you have different cell lines, and at some point in time, you inject the chemotherapy, and you look at the steady state response, or how did that experiment go in terms of the trying to determine chemotherapeutic resistance of certain cell lines?
All right. So with this experiment, we were not trying to apply the chemotherapy during the experiment, but we had a cell which is already resistant to the chemotherapy. Yeah.
So trying to parameterize the conversion rate in those cells as a way to say, like, oh, we have a marker, we can identify a priori certain cells that seem to be resistant or.
Absolutely.
Okay. Great. Thanks so much. I appreciate it.
You're welcome.
All right. I have one more question. I saw your KPL data, which I found very, very beautiful. And then you went to PCA. So I was wondering, does that also then include looking at your pyruvate hydrate? Do you even see CO2 or bicarbonate? And do those add information to what you're looking at to tell something about your cell metabolism?
So here, we didn't see anything like a CO2 or any other metabolite apart from lactate due to maybe low signal to noise ratio. So we didn't have the sensitivity to be able to measure other metabolite like CO2 apart from the lactate. So with the principal component analysis or with the KPL construction, what we were considering was the quantity or the concentration of the cells being able to perform these metabolites. And so all these parameters like the cell numbers, like the concentration of the pyruvate, like the polarization, all these goes into normalization of this downstream metabolite from which we can obtain the KPL constant.
Yeah, thank you so much. Let's thank our speaker again.